Hello and welcome to the sixth webinar that VitaSite has sponsored on islet isolation technology. A few housekeeping things before we get started and make my introductions of Klearchos. During the presentation, please use the chat to ask any questions you have. We will review the questions at the end of the webinar. You can write the chat to everybody or to the admin. It's your choice to whether you want to have everyone see it, but I'll read the questions at the end of the webinar to have Klearchos address them. There are three polls, I should say, that we ask you to complete if you have time. A short poll at the very start of this webinar and two short poll questions at the end. So without further ado, I'll give you some background. I think as many of you know and was in the abstract of the presentation, a big issue for islet transplantation is potency testing. Of course, this is more of an issue in the US than outside the US, where in essence, islet transplantation is handled like tissue transplantation. However, this isn't to say that this is an important issue. It's a critical issue, and we're very pleased and happy that Klearchos has agreed to give a presentation on this topic. Many of you know Klearchos. He's been, if you will, carved out this space and is unique in his expertise in applying engineering principles to trying to study islets. So without further ado, I'll just hand it over to Klearchos. Again, thank you, Klearchos, for doing the presentation.
Thank you, Bob, and thank you for this kind introduction. It's a pleasure to be here, and I look forward to sharing some of the work and my thoughts on this topic, for which I worked for many years since the 90s, actually. I need to restart the presentation, actually, and I'm not sure if how to get to the beginning. Let me just, okay, here we are. Oh, nope. Start. And I'm somehow starting from the summary, even though I want to restart the presentation. I'm not as familiar with this software. I believe I need to go in the beginning. I don't see a button for going in the beginning. Apologies, but I will try and do this manually as quickly as I can.
So yeah, I think that's start. The best.
It may take a few seconds. Apologies as I go through the slides to get to the first slide. But the idea today is go through some of the challenges and limitations of characterizing, especially human islets prior to transplantation. I give an overview of what has been done in the field, and then provide some of my thoughts and suggestions on future approaches that could further improve on what we have today. I also will describe a little bit some of the limitations that relate specifically to the nature of the islets, being spheroids of cells or aggregates of cells, and some of the molecular aspects of the beta cell and the islet that actually make it even more challenging to characterize. So I managed to actually get to the beginning of the presentation. And since we already discussed the title, I will move into the next slide, which is another important slide that describes my commercial interests. I am a co-founder and stakeholder in Procyon Technologies LLC, as a startup out of the University of Arizona that works on encapsulated cell therapies with the focus on type 1 diabetes. And move on to the next slide, with which I'm summarizing the key items we are after, or the goals, when we are looking into pre-transplant islet quality assessment. And for a given islet preparation, one key question is, perhaps the primary one, is this safe to transplant? What is the purity of the preparation? And of course, then what is the potency of the preparation, or the dose, if you will, that you're planning to transplant? One example of that is the number of viable functional beta cells per kilogram body weight of the recipient. And can we predict really the transplant outcome using a set of real-time assays, meaning a set of assays that we can complete and get the results prior to making the decision to transplant a preparation? I'd like to set the stage by providing some background on islet characteristics and also some mechanistic information about how the islet works, which may be, or the beta cell works, which may be important as we characterize these preparations. And I think it's a factor in the complexity of assessing islets prior to transplantation. In the pancreas, the islets are embedded into the tissue. They're highly vascularized, and of course they contain not just the beta cells, even though the beta cells are the majority. The insulin secreting beta cells are the majority of the cells within these islet spheroids, but alpha and delta cells are important, and there's of course a network of communication within the pancreas. So it's very important, and of course that's disturbed and distracted when islets are isolated and purified and cultured. This is a little bit of more of a focus on the islet microvasculature, very complex and very important for its function. This provides a high capacity for exchange necessary for islet function, exchange, for example, of nutrients, oxygen, and hormone dispersal in a very fast way. There was quite a bit of a challenge originally in the field in how to count or quantify islets. There are cell clusters that can range in diameter from 50 to 400, sometimes more. So a decision was made by some of the leaders in the field to normalize and define an islet equivalent as the volume of a sphere of 150 microns. And that represented the majority of this, the islets, the size of the majority of the islets, for example, in rat islets and also in human islets. So that was a reasonable way to represent and define the islet equivalent, and gave a way to normalize when counting. And an islet equivalent can have about 1,500 to 2,000 cells. And that actually can vary depending on the vasculature state inside it, because that's a volume measurement. And an example is that if the vasculature of the islet collapses, as you saw, it takes a very significant space within the islet, maybe 15 to 17% of the volume could be the vasculature. If it collapses, the volume will change, but the number of cells will not. So when you look at that, and the time at which you look at that could affect the number of cells you're actually measuring in that volume. And the majority of these cells are beta cells, but as indicated earlier, there are also alpha cells which produce glucagon, a very important hormone, and delta cells that produce somatostatin. So we need to think about it as we characterize the islet preparations, and I'll elaborate on that in later slides. Another important aspect is that the islet function, or the beta cell function, is highly sensitive to hypoxia. And that is another important part. There are several people who have done work on this, even in the late 60s, Klaus Hellerstorm and other European groups. It's also with perifusion work, the group of Dr. Colton at MIT published a paper showcasing the kinetics of this effect and how dramatic it can be, especially for the second phase or the glucose stimulated part of insulin secretion. And I don't have a pointer here, but if you can follow the trace of oxygen as it goes from about 140 millimeters mercury, which is essentially incubator air saturated, as that declines, you can see the effect that is very acute on insulin secretion. And also you can see a prompt recovery upon reoxygenation. So short-term effects can affect short-term exposure to hypoxia, let's say minutes, can have a very acute but reversible effect on insulin secretion. Another important point from this paper is that even if the oxygen concentration was reduced to around 30 to 40 millimeters mercury, as you see on the right panel of this figure, the effect on insulin secretion is still very substantial. And the interesting or important point here is that oxygen level is the level that is available at common transplant sites. So even if you had essentially every islet exposed to that concentration at its periphery, at its surface, which is what happens in a perfusion chamber, you still had impairment in insulin secretion. And that's a very important part, and it does relate to transplant, because even if you had the most functional and happy islets, they would still be limited, and their function would be limited in the absence of any other problem. They would be limited by the oxygen that is available at the transplant site. Of course, that's dependent on size and other factors, but this can serve as a warning, especially as we try to correlate or relate pre-transplant assessments to transplant outcome. So by now you probably have appreciated the importance of oxygen on function in the acute exposure, but it appears that also long-term exposures or longer-term exposures could affect function to a greater extent than viability. And there are two important aspects of this. Beta cells are not really able to effectively produce ATP, essentially energy, to support them under anaerobic conditions or low oxygen conditions. They have a very low expression of lactate dehydrogenase, an enzyme that can convert pyruvate to lactate and provide energy in the absence of oxygen. And this enzyme is present in tumor cells and other cells that are capable to do that. And this has a lot to do with the way the beta cell function and if beta cell functions and also the beta cell senses glucose to release insulin. And if you overexpress that enzyme so that you enable the beta cell, let's say, to produce energy under low oxygen conditions, you essentially impair insulin secretion. There's some very important work done late 90s on this topic, and it's a good read for people who are interested in that. More recently, other groups have reported a pre-existing hypoxic gene signature in particular human islets, perhaps human islets that were obtained from organs that were preserved suboptimally or were not ideal. And that correlated with impairment in function. And that actually impairment of function appeared to be persistent, even though the exposures were relatively short. So that's another important point. And we also recently reported with human islets that exposures up to 24 hours to extreme conditions of, let's say, ischemia, essentially pelletizing, putting human islets in a pellet over 24 hours, had a pretty drastic effect on viability, but there was still a remaining fraction, perhaps 33% of the cells were viable. But when we looked at the same tissue for function, and here is the perfusion, and red here you see the normal islets that were exposed to normoxic conditions, a nice first phase and response, and a second phase sustained insulin release. However, when we did the same on the remaining cells with some remaining viability, let's say 33%, they were essentially flatlined instead of function. So the function was dramatically impaired, even though, again, there were viable cells remaining. And of course, when we looked at the molecular profiles, we definitely saw similar things as were reported by other groups, where we had the HIF1 alpha pathway and the TNF pathway, inflammatory pathways dramatically affected and activated. And actually, the pro-inflammatory pathways may be being activated under such exposures could be very important for transplant outcomes, especially when you're dealing with allotransplantation. So some things to consider when thinking about characterizing preparations and how they can predict or not, the assays can predict or not post-transplant outcome. The hypoxia and ischemia exposure is very important because, unfortunately, there are a lot of steps during which islets, human islets or otherwise, could be exposed to such conditions, starting from the donor, actually, brain death, perhaps some warm ischemia, but also cold ischemia during preservation, stresses during islet isolation, repeated pelletizing, and potentially during islet culture and distribution. So there are multiple points at which the islets are exposed, whether they're in the pancreas or isolated, to such stresses that can actually damage, not just kill them, but affect their function. So it is very important for that reason, and not to mention donor-to-donor variability, to characterize the preparations pre-transplant and also characterize them perhaps at various stages, for example, pre-culture and post-isolation. And ideally, also, if one could, post-transplant monitoring would be fantastic with some imaging techniques and other capabilities. Now, having shared that background, I would like to move into some of the work that has been done in the field then to characterize islets. And as clinical islet transplantation started to become more of a reality with great successes from the Edmonton group around 1999 and 2000, and University of Miami and University of Minnesota and others, there were, of course, important thoughts about standardize the islet isolation technology, and actually the lack of potency tests, viability and potency tests that actually were meaningful was a challenge identified by some of the leaders in the field in this paper here. And that actually served as a momentum for more work in this area, especially as it was recognized also at the time that the FDA, based on its regulations, would require some robust pre-transplant characterization of viability and potency for any approval of such procedure. And I believe that was pretty clear around that time. So, what are why is it so difficult to characterize islet preparation? So, what are the challenges associated with that? One key one is the fact that there are cellular aggregates. Many assays developed for characterizing viability of cells have been developed for single cell suspensions or monolayer cultures. Here we have spheroids of different shapes that are not really perfect spheres. Trying to actually estimate volume in 3D is not trivial when you do it in 2 dimensions. So this can be prone to error, they're operator dependent, and there could be large uncertainty. So the other part is that the human islet preparations have varying amounts of impurities. You start from perhaps about 100 ml of tissue, and you end up with 2 to 3, sometimes more ml of tissue, and you should not have, if you had ultra pure islets, you should not have more than 1 to 2 ml of tissue total. So the fact that there is a lot of impurity in the preparation is another complicating factor. As indicated earlier, the islet can be a moving target. Damage occurs during multiple stages, and I share that, so I will not elaborate on that. The other part is that many of the techniques that are used for cells are really not applicable to islets because islets are not easily dissociated into disparate cells, and when you try to do that, you can damage the cells in the process. Cells get lost, and maybe some cells are preferentially lost in the process. So when you try to assess what's left, you're not assessing what you started with. And the recovered cells may not be representative of the original islet. So that's just some of the key challenges in characterizing islets. It has in the field, it's pretty widely accepted that the nude mouse bioassay is one of the most reliable assays or predictive assays with respect to clinical outcome and characterizing preparations, and there is some important literature on that, and some data available on the web, maybe not formally published, but this is not something that is disputed by many in the field, and some recent work also out of France with Dr. Kerkonde's group correlated clinical outcome with human C-peptide in mice. They found that was one of the few things, or maybe perhaps the only thing, that correlated with the transplant outcome in their hands. Again, consistent with some this nude mouse bioassay or a mouse bioassay being predictive, not without challenges, but perhaps one of the best tools that was available. Unfortunately, this is retrospective, so it takes weeks to get this outcome, and it's not useful in actually prospectively assessing the preparations. The current product release criteria, as described by the field and used clinically, relate to safety. I won't elaborate much on that, on the endotoxin and gram stain. The identity, which basically is an islet count, essentially a volume count of islets per kilogram body weight of recipient, with a minimum number being 5,000 for the first dose and 3,000 for the second dose per kilogram body weight, 3,000 and 5,000 islet equivalents per kilogram body weight. The viability is looked at with a dye exclusion, either FDAPI or comparable. It has to be more than 70%, and purity is measured by dithazone, and we'll elaborate a little bit more on that, and it has to be greater than 30%. And the potency is measured by glucose-stimulated insulin release, which is a current static assay, not a dynamic assay, and a stimulation index of greater than 1 is enabling the preparation to be transplanted. And stimulation index is really the ratio of glucose-stimulated insulin secretion over basal insulin secretion. So theoretically, when combined, information on this identity, viability, and potency, as described in this table, should provide information on the number of viable and functional beta cells transplanted per kilogram body weight of recipient, and should be predictive of transplant outcome in the absence of immune rejection. Now, that's again if you combine this information. None of this by itself could be really predictive without the other information. And that's an important note. So, for example, measurement of the islet equivalent number, and that's also true only if these measurements are accurate and precise. And for example, measurement of the islet equivalent number, as stated earlier, and the viability and purity, it may not be for the variety of reasons that were described. And of course, if your beta cell fraction were relatively constant in human islet preparations, or it's measured and accounted for, then it could be used. But again, there's great variability in that in preparations, and I'll show some of published data on that in later slides. And of course, beta cell function, insulin secretion, is not impaired or is within acceptable limits. And again, the stimulation index is not an ideal measure of that by itself. I already went through the challenges and limitations, for these assays that are already existing. It's great that we have them, and they can be very helpful, but there is quite a bit of room for improvement. I will not go any further on this slide, except to see that perhaps elaborate or built on what I stated earlier, that the stimulation index, which is the ratio of glucose stimulated over basal insulin secretion in a static incubation setting, by itself cannot be predictive of transplant outcome. So it's really not appropriate to try and correlate a stimulation index with transplant outcome by itself. And there are some people who've tried to do that. Just one quick example, you can have 5 islets that have a stimulation index of 10, and they're amazingly potent. But if you only put 10 islets into a patient, there's no chance for diabetes reversal. So you have to at least have some marginal dose associated with that. So very important to actually think about what each measurement does and use it in the appropriate way. It can be proven more useful when combined with information such as specific basal and glucose-stimulated insulin secretion, meaning normalized per unit cell or per beta cell or per DNA of beta cell or per nucleus of beta cell, and even better if it's obtained in a dynamic perfusion assay. It's also maybe very important to consider alpha cell function and glucagon release. That is something that perhaps could be considered in the future. So what are the characteristics of interest and what tools do we have available? I alluded to the volume earlier, or islet equivalent, the number of cells and the composition, perhaps the fraction of alpha cells and beta cells in the preparation or other cells that are present. Membrane integrity, mitochondrial function, apoptosis measurements for viability, and of course for potency, the glucose-stimulated insulin release, static or better yet dynamic with perfusion, and the retrospective assay of the immunodeficient mouse transplant model. The quantity of the tissue is currently characterized by packed cell volume or tissue pellet, and the islet volume is characterized by visual counting and enumeration of the islets in 2 dimensions under the microscope, utilizing dithazone staining, which stains specifically, binds to zinc into insulin granules and can stain specifically islets. Again, a challenging method to be quantitative given the preparations. And the cell composition, again, purity is measured by D1000. It's really not a cell composition assay. Now, improved test methods for that could be the use of DNA and nucleic counts for the entire islet preparation. And then, of course, normalizing or accounting for beta cells and alpha cells using other techniques such as immunohistochemistry or electron microscopy to characterize the fraction of beta cells. So then you can get a better estimate of the fraction of beta cells relative to total cells in the preparation. So those would be improved test methods that could be very, very useful. And I'd like to highlight here Dr. Danny Pipeliers and his team has spent a lot of time and energy, done a great service to the field by characterizing islet preparations in that way, very quantitative assessment of beta cells and alpha cells, and certainly try to correlate that with function and also transplant outcomes. Another important part of this is the variability in the preparations, highlighted especially in the differences between human and rodent islets, highlighted by Dr. Power's group in a paper published more than a decade ago. But here you can see the large variability in the beta cells in an islet population, going from, let's say, close to 30% to close to 80%, depending on the preparation. So that's an important reminder that there is a lot of heterogeneity in the preparations. That was even further highlighted by Dr. Ricorti's group with a paper published by Dr. Ichi. And there, the beta cell fraction, they actually looked at purity by D1000, and on the top right, you can see the x-axis. Again, unfortunately, I don't have a pointer, but if you look at the x-axis and you go down to the 90% purity, as measured by D1000, you can see the beta cell fraction in that preparation. I highlighted this into the slide with the circle. You can see the beta cell fraction can vary from 15 to 45%. Again, for preparations that we deem 90% pure. So that shows you a little bit about the importance of measuring beta cell fraction and not just relying on D1000 for the purity. And we also have done some measurements with clinical islet preparations, published data that in terms of volume measurements, the purity is definitely overestimated. For example, for the highly purified preparations, 70% pure and above, we had volumes that were measured that were ranged from 1 ml to more than 4 ml, whereas the predicted volume, if you had this purity, would be substantially lower. And that effect was even more pronounced when you had the less pure preparations. And again, this is the slide, the figure B here. And importantly, the viability as measured by both oxygen consumption to DNA and the less sensitive membrane integrity test was lower consistently in cultured islets when they were less pure. This is from the same preparations. So you had a pure fraction and less pure fraction cultured, and the more pure fraction had a higher viability than the less pure fraction, suggesting that culture of less pure fractions may actually further damage and perhaps more than the culture of the pure fractions. Another important consideration. So for the viability, the current standard is a membrane integrity test. An example is the fluorescent acetate from Viability assay, and there are many others. Unfortunately, that is not a very sensitive assay, and especially when it's used in the 2D as opposed for three-dimensional objects. Very hard to look at the interior of the eyelid and see and quantify, not say quantify, what's dead and what's alive. And that is only counting cells that have a compromised cell membrane. It does not count cells that are in early apoptotic stages or have not yet completed the death cycle. Another assay that is used, or other assays are based on mitochondrial function, and this seems to be a lot more predictive and also a lot more capture a lot more modes of death, including early apoptotic events. So these assays could be very useful as moving forward. And now I will move into the last part of the presentation, where I will elaborate a little bit on some of the attempts for prospective assays, especially ones based on mitochondrial function, and to predict transplant outcome in the nude mouse bioassay, which was again the most predictive assay available at the time for clinical outcomes. So this was be used as a bridge, essentially, to define prospective real-time assays that could predict that outcome, that could then be used to predict clinical outcome. A lot of that, what I present, is focused on work we have done in collaboration with many other groups over the years, but there are other people and other groups working on this, and I apologize if I cannot be comprehensive here, but I will focus on our experience primarily with these assays and this presentation. So the hypothesis is that if the oxygen consumption rate per viable cell is relatively constant, and there are a lot of good reasons to believe that's true, and it has been also utilized in large-scale bioreactors for protein production and other work. So this is not an unreasonable assumption, and again under defined conditions. But the oxygen consumption rate, if that is true, the oxygen consumption rate can be proportional to the number of viable cells or the volume of viable tissue in a preparation. And in more simple words, the amount of good tissue in the preparation. Now, the DNA can be a measure of total cell number or total tissue volume, and that can be very quantitative, and that is a representation of the total amount of tissue. So the ratio of oxygen consumption rate to DNA it can be a measure of the amount of viable tissue to the amount of total tissue in a preparation, which is then a measure of viability or the quality of the tissue. So if this is a reasonable hypothesis and assumption, then if we assume that the function of beta cells is not impaired, then the viable beta cell number or dose per kilogram recipient body weight would be expected to predict transplant outcome in the absence of immune rejection. And that could be better than the total cell number, because it actually accounts only for the viable. For example, total islet equivalence would not account for the dead cells if we didn't multiply that with a measure of viability. So we tried to relate that to the nude mouse bioassay, and we published a paper that was very collaborative with the City of Hope and several other centers that participated in this and sent human islets to us at the time at the University of Minnesota, where we would characterize them by OCR and OCR per DNA, and then use these parameters to predict transplant outcome. And we showed that when you combine the OCR dose with the viability measurement, the OCR per DNA, we have a very good, very predictive outcome with respect to nude mouse bioassay. But we needed these two parameters here, not just the dose of the viable islets or cells, but also their viability. And this is something I believe specific to the kidney capsule, and the model was used in which the presence of dead tissue actually affected the survival of what was viable tissue there. So it is a very interesting cautionary note that maybe the kidney capsule has some specific limitations that need to be accounted for when you're looking at predictive assays. And that was further shown, and actually it was shown that the OCR was predictive as a dose of the marginal dose required for diabetes reversal, and that marginal dose was highly dependent on the viability. So that's another interest. So the presence of dead tissue here affected the marginal dose required of viable tissue to reverse diabetes. Maybe specific again to the mouse model used, but that's very, very interesting and important. Now, quite a bit of additional work was done in the field by many other groups, and I'm just highlighting some of the papers here. There's some more recent work as well. And I would urge anybody who is interested in this to look up these papers for some very important reading. Now, as the last part here is attempts to relate and predict clinical transplant outcome using these assays. And we were very fortunate to be able to work with Dr. Melina Bellin and Dr. David Sutherland and Dr. Herring at the University of Minnesota in the clinical auto-transplant setting in the liver, where primarily intraportal transplants, where we related the OCR dose, essentially the viable cell number per kilogram body weight of recipient, to the transplant outcome, insulin independence versus insulin dependence in auto transplants. And here you have a scenario where you don't have autoimmunity to worry about, you don't have alloimmunity, and also the pancreas preservation is rather limited. So it's a much simpler setting than the islet allotransplant setting. And also in this setting, you essentially transplant any islets you get from this donor, who is also his own recipient. So there's some variability in dosing. So here in this setting, the OCR dose by itself was highly predictive of transplant outcome and better predictive than just the islets alone. And there's another interesting observation. And here we did not use a need, essentially the second parameter of OCR per DNA by itself to be predictive. And OCR per DNA by itself, as the viability by itself, as shown here and as expected, was not predictive at all of transplant outcome. It had to be combined in order to produce the OCR dose or the viable cell dose to be predictive. We were also very fortunate to do some limited work through the CIT, through some of the NIH-funded phase 3 trials with Dr. Shapiro and his team. And with a limited number of islet allotransplants, we're also able to show that the OCR dose was highly predictive. With that, I'd like to also acknowledge the funding that, at least for our team and collaborators, helped in getting this data, especially the preclinical and clinical work, and summarize the progress made and perhaps some suggestions of future development. So while great progress has been made in the field, I do believe there is a need to further develop and refine real-time predictive potency tests for clinical islet transplantation, especially for further optimizing also the process. Islet nucleate counts and DNA measurements may further improve islet dosing, especially when combined with measurements of beta cell and alpha cell fraction. Measurements of islet preparation purity should be further refined, and the relationship between islet purity and transplant outcome should be further explored. I think that's very important, given some of the information we shared and the data out there. And viability and potency assays based on mitochondrial function, for example, oxygen consumption rate, appear to be useful and should be further explored. But people should not assume that just one parameter can do it all. And for example, OCR per DNA, a measure of viability by itself, cannot be predictive of transplant outcome. And then attempts to correlate the clinical outcome should take into account viable and functional beta cell dose. With that, I'd like to acknowledge my team members and also collaborators for all their contributions. Thank you.
Great. Thank you. Let me turn on my mic. Can you hear me? Yeah. Thank you very much, Klearchos, for the webinar.
You're welcome.
Excellent. Excellent. There's a number of questions. I just signed on again, and I know there were a number of questions that I don't see anymore. I was wondering, do you see the questions? Because I had to, somehow I got a glitch in my internet. I had to go back to a wired system instead of the Wi-Fi. So could you answer the questions, Klearchos, do you see there?
Yeah, I could try. Yeah, I see.
Because I know that Wang Si Sui answered some asked some questions, as did Hannah Schultz. I don't see them anymore, unfortunately.
Yeah, I see a lot from Dr. Xeng, and there are great questions. They are pretty detailed. I see. So should we start from the?
Yeah, start from the first. Yeah.
So I see
Hannah Schultz.
Yeah. Bob, will you be reading them, or would you like me to read the question?
No, I don't see them for some reason. I got a glitch in my internet connection, so you'll have to read them. I don't have them on my screen anymore for some bizarre reason.
Yes. So I see the first one from Dr. Stu Williams. Great question. So his question is, any thoughts on how we can perform these types of assays in our autologous islet transplants? Many times these islets are heavily embedded in acinar tissue. That's a great question. And Stu, in the paper we published with Dr. Sutherland, Dr. Belling on the clinical islet transplant, auto-transplant outcomes, we actually initially did the measurements on just the highly purified fractions. We utilized highly purified fractions, but then expanded this to impure, heavily impure fractions. And we can measure the OCR from the heavily embedded islets and the pure preparation. And we also measured the OCR on the ultra pure islets and the acinar tissue, and found out that it was very similar. So then we actually used a purity estimate to calculate the OCR specifically of islets from that. And I know this may sound complicated. I'm happy to describe this more offline, but we were able to still be very predictive using this and assuming essentially that after measurements that the OCR of the acinar tissue is very similar to the OCR of the islets, and just accounting only for the islet fraction in that, based on a good estimate of the purity. I would be a lot more comfortable, and it'd be a lot better if you had beta cell fraction with a quick assay there to account and essentially multiply the fraction of beta cells or islets with the total DNA or total nuclei count of the preparation and calculate the OCR of the islets from that. I hope that satisfies, but I'm happy to have an offline conversation on this. And Dr. Waxing has 3 questions. Let me see if I have. Let me see here. I see another question actually before that, Dr. Julian Tevenet. Is there a defined threshold for OCR to discriminate good or bad islets? So we actually have some information on that in the published papers, at least for the mice. If we had an OCR per DNA between 150 below 150 nanomole per milligram per DNA, and we did not get ideal results. A range between 75 and 150 gave us mixed results, but you could actually account for that by increasing the dose. And if you had anything below 75, you generally got very, very poor results and never really reversed diabetes in the mice. It's not as simple as that, but hopefully that gives you some idea there. And then Dr. Kui asks, do you think the FDA should treat the human islet product as a drug? If yes, please explain the reason. If not, what is your opinion? Yeah, I'd rather leave this up to the FDA, but I will say that in my opinion, it's very important to have predictive assays and quantitative real-time assays that are meaningful prior to releasing islets for transplantation. I'm a firm believer in that, and I also believe that helps the field by trying to optimize the process, whether it's with enzymes you use, whether it's with organ preservation method you use. So I do believe it's very important, but I will not comment on how this should be treated. I think the policies are there, and there is a very good and robust debate in the field about it. I'll leave this to people who have a lot more experience along those lines. Then the other question was, do you think the GSIS assay can fulfill the FDA potency testing requirement? For allogeneic pancreatic cell products. I don't believe the static potency test, GSIS test, is sufficient. But if it's combined with other quantitative measurements of beta cell number, beta cell viability, could be potentially good. I would prefer something that is dynamic, like the perfusion, because there are some limitations with the static assay. So by itself, not, but when combined, as I indicated, with other measures, potentially yes. And yeah, I think there's a lot more here on the FDA, which probably requires an offline discussion. Otherwise, I think we'll be out of time. So thank you so much for your questions there. And then, so I'll be delighted to have an offline conversation, Dr. Kui.
Yeah. So.
And then the last one, how will you suggest to measure the alpha cell function in isolated islets? So that's an interesting question. I mean, we have, and others have done some work in the perfusion assays, where you look at glucagon release, and there's some multiplex assays that are available. So that could be one way, but I'm sure others we have other suggestions. And Dr. Kerkont here, I don't think she has a question, but she has no stress. So I don't know if that's no stress to islets or no stress.
That was at the very beginning when we had problems with the slides, Klearchos.
All right.
So in other words,
I think I covered all the questions. I'm sorry, it has been a little challenging just to read through everything.
Yeah.
Hopefully I covered it.
Yeah, I really want to thank you. I just want to ask one question. I know you in the past have worked for trying to oxygenate pancreas to preserve what you've shown very elegantly in the presentation here. What is the status of that now? Anyways, they had the two-layer systems and... Yeah.
Yes, great question, Bob. And we did publish a paper actually in 2019, I believe, with the collaboration with multiple centers, and that actually showed that oxygenation of the pancreas is extremely helpful in maintaining functionality of the islets, and also we looked at that at the molecular level. So if you oxygenated the pancreas during preservation, you had lower pro-inflammatory and hypoxic signatures in the actual isolated islets, so which I think is very impactful. So we unfortunately didn't have. We did a lot of work to develop equipment that would allow us to persiflate, and that didn't really materialize, at least into a clinically relevant or usable unit or set of units. So it was experimental, and so it was difficult to then pursue it at the larger scale. But I believe Dr. William Scott over in Newcastle has worked on a simplified system that could be clinically available, and I believe this could have extremely positive impact if used and verified further for human islet isolations, because I do believe the hypoxia or ischemia actually during preservation begins a process that damages islets both in terms of functionality, but also viability and perhaps minimizes the yield. So I think there's a great opportunity there to either that or other techniques to improve yield and quality of the product.
All right. Well, thank you again, Klearchos. This was fantastic, the way you've organized this very extensive and complicated science. And I'm grateful you could take the time to do this webinar. Just want to remind people, if they get a chance, just to answer the last 2 poll questions, and we will have another webinar next quarter. So thank you again for all your participation. Thank you again, Klearchos.
Thank you, Bob, for inviting me, and thank you, everybody, for your questions. And I'll be happy to speak to any of you offline if that is allowable. And I do look forward to seeing islet transplantation getting to patients here in the US as well consistently. I think that's very important. Thank you.
Thank you.
Thank you.
So long.
Bye-bye.
Bye.